Hi, everyone. Thank you for participating in the Spatial Omics University 6. And today we are very excited to have Dr. Ying Zhu from PNNL. And of course, long version of PNNL is Pacific Northwest National Laboratory. And briefly, Dr. Ying Zhu received his degrees from Zhejiang University in China. And then he also worked at the National University of Singapore in the Department of Chemical Biology. And after that, he worked at the ETH Zurich as a visiting professor. And also during his time at the Zhejiang University, he was also an associate professor. After that, he started his career at the Pacific Northwest National Laboratory since from 2016 as first postdoctoral researcher and now as a senior research scientist. And they're also part of the NIH HubMap program, where they are currently working on single-cell protein analysis technologies, which they call NanoPOTS. And without further ado, I'd like to turn over to Dr. Zhu, please.
Thank you, Ahmet, for the introduction, and thank you for your invitation to this seminar. So I personally really like this seminar. I always join every Friday to learn all the new progress in this area. So today I'm hope to give some overview on what we are doing in PNNL to developing mass spectrometry-based single cell and spatial proteomics technology. So as we know, single cell transchromatomics really transformed biology field in the past 10 years. And actually single cell proteomics, actually the concept come out very early, about almost 20 years ago, but there's a lot of challenging and to achieve measure protein in single cells. That's why majority of the current single cell proteomic approach is based on antibody, like mass cytometry or other macrofluidic approach. So we are working on actually using liquid metalography and mass spectrometry to directly measure all the protein without using any affinity approach. So to start, can you see my screen now? Okay. So the first question is really, do we want to measure proteins? As you can see, this very interesting publication published on Nature 10 years ago to study the two different modality of mRNA and protein in the human cells. As you can see, they're quite different. They have very different half-time. mRNA usually 9 hours, but protein can stay much longer in the cells. And the copy number are totally different. RNA very low. Usually the median of the copy number is 17, and for protein is much higher. We can get a median of the copy number 50,000 for proteins. And there's a very low correlation between the half-time of the protein and the mRNA. That's maybe the tell us why and the correlation of between transcripts and protein is quite low. If you look at here, the correlation is only 0.4. It tells us maybe only 40% of protein can be predicted by gene. It's also a very good justification to for us to derive measure protein. This actually is a recent publication by my participant group. They use a new technology to measure protein in single cells. It's also mass spectrometry approach. And then at the same time, they also use two different RNA sequencing, SMART-Seq2 and DROP-Seq to measure the RNA abundance. So if you look at the PRC correlation between the three different measurements, they're quite high at each modality. But if we do a cross correlation between protein and mRNA, is quite low. If you look here, it's usually at much lower the bulk scale. It's usually 0.2 to 0.3. It also tells us it's hard to use RNA to predict the protein. So so the field actually is waiting for the technology. It's also quite interesting to look at how the natural technology future. If you look at 2 years ago, the name of the topic is the dream of single cell proteomics. It means it's your dream 2 years ago, and people don't know if it will be true or not. And now, just 2 years later, the natural editor published another technology feature is just come out. The title turned to single cell proteomics tech center stage. So it's clear tell us the field is going very fast in the last 3 or 4 years. What's the major challenge of single cell proteomics? At first, we know for transcript or genomics, we can use the PCR or other amplification approach to amplify the molecular to get a very high number of molecular to do the measurement. However, for protein, there's no such approach. You have to, what you have in the cell is what you have. So you have to have very sensitive instrument to measure them. And if you look at here, the protein combinatorial distribution here is also, if you want to measure 100 protein, maybe it's fine. I think most mass spectrometer can measure 10 atmole molecular. However, if we want to go to 5,000 or 8,000 protein, we need a very sensitive mass spectrometer. We need if you look at common number is 10 zimmeltomole, it's just 6,000 molecular. And let's take back to look at the procedure of a current proteomics approach. Usually proteomics use 3 major steps. The first step is sample preparation. So we need to lyse the cell and extract the protein from the cell. And finally, do some chemical modification like reduction or alkylation, then use trypsin to digest the peptide, digest the protein into small peptide. The reason we do the digestion because the mass spectrometer is more sensitive to measure the small peptides. The second step of proteomics is use the liquid chromatography mass spectrometry to separate all the very complex peptide mixture, and then use the electron spray to ionize the peptide from the solution phase to the gas phase. So unless that we go to the gas phase, mass spectrometer cannot measure them. So after collect the gas phase ions into the mass spectrometer, then we can select each peptide to the fragmentation to break down the peptide to small fragment. Then we can use the database search to align the fragment to the theoretical fragment of the peptide to the measured peptide to the peptide identification and then to quantification at the protein level. So if we look at the state of the field back to 2016, this is actually the best data for a small number of cells. It's published by a group in China, and they're developing the new sample prep approach to allow to analyze low number of cells. Here you can see the lowest number of cells is 2,000 cells. Not single cell, but even use 2,000 cells at the 2016, it only can identify about 2,000 proteins. It clearly shows the gap. Is a huge gap to measure single cells. And but if we look at back the mass spectrometer raw sensitivity, so here is the publication on mass spectrometry journal. It's by Alexander Prof. Is actually the inventor of the new detector. It's called OB-Trap. It's a high resolution mass spectrometry detector. In this paper, he showed actually use OB-Trap detector. We can measure a single molecular. Here is not a peptide, actually is a single myoglobin protein. It's an intact protein. Part of the reason it can measure a single molecular protein molecular is because each myoglobin, each intact protein actually carry multiple charges. That's why it have better sensitivity. actually the orbit travel is a charge detector. The more charge is the more sensitive. Anyway, it shows the mass spectrometer can detect very low level of signals and as low as like 10 charges. In our lab, it's also published very long time ago, is 2004. It's almost 17 years ago. We show use the liquid chromatography and mass spectrometer. We can measure a very low number of BSA digest. And so as you can see, we can detect 75 zivtomor BSA peptide use RC and MS. So by calculate our ROD, actually we can measure 10 zivtomor is 6,000 molecular. It all tells us the mass spectrometer RC is have sufficient sensitivity to measure the protein in single cells. So, but the reality at that time is we still need 1,000 in like 2,000 cells to do to get 1,000 proteins. So the major change we realized actually is between the analytical sensitivity and the reality is the sample prep. As we know, protein is very sticky. There the non-specific binding to all the surface, most of the surface, and all this proteomics procedure we are using before is require lyse the cell and do all the reaction and digestion in one vial and do the purification, use some column and transfer to RC. So all the procedures, if we use a very low number of cells or one cell, we just lost all our peptide proteins. It's a problem of sample recovery, is not a problem of the mass spectral sensitivity. So that in realize this challenge, that's when I come to PNNL, so we are working together to developing a microfluidics technology. We call it a NanoPOT. It's a nanodroplet processing in one pod for trans samples. As you may see, the microfluidics design is quite different from other microfluidics approach. We do not use any channel or the closed channel. What we do is just pattern the surface of a slide, it's glass slide, with hydrophilic, generate hydrophilic spot on hydrophobic background. So it's actually similar to the early stage of microarray. So we do all the reaction in the open space in small droplet. As you can see here, we do all this sample prep in one droplet. We usually at the beginning we use a total volume of 200 nanoliter droplet. So we do everything inside the single droplet, like including sample isolation, extraction, digestion, and collect into the LCMS for analysis. And so we also built a robot to do all of this because there's no commercially available at the time. So this actually was the advantage of small droplet for the sample prep. If we look at the volume, we can reduce the volume almost 1,000 times. And the most critical one is actually the surface contact area. If we use the tube approach, it's a typical 100 microliter volume. The contact area of the tube is over 100 millimeters square. But if we use the nanopod, we use the one droplet, the surface contact area is less than 1 millimeter square. We can achieve 99% reduction of the surface. So that's why we can increase the sample recovery during the protein digestion. And also very critical one is also digestion kinetics. We all know the trypsin digestion is concentration dependent. So if we do all this single cell digestion in big tube, the protein concentration is just too low. We cannot get sufficient kinetics to digest the protein into peptide. If we minimize all the reaction in small volume, as you can see, we increase both concentration of trypsin and protein. We can get very efficient trypsin digestion. And the first demonstration of the technology, we didn't use single cell. We use serial diluted cell. It's cultural HeLa cells. And we just get 10 HeLa cells in the 1 nanovirus, 100 HeLa cells. As you can see, this is the typical LCMS data. So even use only 10 HeLa cell, we still can get sufficient, very good signal here. If we look at how many peptide protein we identify from the 10 HeLa cells, usually we can look just at the red bar. We can get a 3% of a protein from only 10 HeLa cells. And we also did a comparison, had a comparison use the 25 microliter in and panel of vials. Even use a low binding one, still we can only get less than 500 protein from 25 microliter sample prep. It's again shows if you want to get a high sensitive protein measurement, we have to use a very small volume to increase the recovery. And we next just try to do single cell proteomics. The first trial actually just coupling flow cytometry with the NanoPOTS, as you can see. Because maybe you can see the nanovirus is actually not very small. The diameter of the wire is 1 millimeter. So that's why we can use flow cytometry to sort the single cell into this wire. Use this further demonstration, we measure different cell number. As you can see, even use one cell, HeLa cell, we can get identify 700 protein most from single HeLa cells. And usually for quantification, we require not only we require at least usually we require at least 2 peptides in more confident identification. So we still can quantify over 300 protein from single HeLa cells. And it's the first study to use the lab-free single cell proteomics to study the cultural cells. We next try to evaluate if we can use single cell proteomics to detect the perturbation of the cultural cells to see if we can see the cell state change. Here we use a well-known model system is macrophage activation. Is use mouse raw cells. We treat the mouse raw cell use LPS to activate the immune response. We treat at 3 different time points. 0 hour is at the control, and 1 day and 2 days. So after measurement, so as you can see from the UMAP projection, so clearly we can see the cell is totally different after treatment. Even at 2 different time points, it's also so the protein profile of the single cell also show that they have a different response at 2 time points. If we look at the some marker protein and the immune response marker protein, also we can see there are very low level expression at the control time point 0. They get abundance get higher at 24 hours and 48 hours. Also, we can see some protein actually after the long time treatment that just go up. It's a kind of early response of this protein. The level go higher at 24 hours, but go down at 48 hours. So we also collaborate with Peter Gispi at OHSU in Portland. So his biology interest to want to understand how hair cell, which is the cell is mechanical transduction cell, is to transform the sound into electrical signal. The major challenge of the hair loss is the hair cell cannot regenerate it. And so the major focus on his study is try to understand how cell differentiated from progenitor cell. Here we call it a supporting cell to hair cells. So as you can see, we use one dye is FM143 and to specifically label the hair cells. So we can sort hair cells, and then we also can sort the supporting cell into our nanopod device, and then do the protein profiling. And use the protein abundant information. Actually, we can generate kind of pseudo time from supporting cell to hair cells. And to actually, we all find out all the marker of the hair cells and supporting cells. Very interesting, we see one protein is TMSB4X. It's a very small protein, and it's a actin binding protein. We see in protein level, we see it have a same level of actin. Um, um, the reason we have interest to actin because this bundle actually is the protein bundle, it's actin bundle. It's actually this bundle transform the sound into the electrical signal. It's have a very similar number, common number to this protein. That's why it gives us a new hypothesis. Actually, in supporting cell, all the actin is already expressed. They just do not release to build the bundle. So that's what see is during progress from supporting cell to higher cell, you see the TMSB4X degraded. So during the degradation of the TMSB4X, it release the actin to build the bundle. So we also use immunohistochemistry to validate all the proteins. They're very high in supporting cell, very low in the higher cells. So even we see this study makes sense and we can show single cell predict can really give us a new biological insight, but still a lot of challenge here is, as you can see, we use LCMS. Each LC require at least 1 hour of separation to get one cells. So even at a different gradient time, we can see we can measure only maximum we can measure 24 cells per day. So it actually is very high cost if we want to measure a lot of cells. So that's why we try combine another technology developed by Nikola Slavov in Northeast. It's called Scope MS. And the idea is just using the tandem MATEC is a reagent commercially available by Thermo Fisher. It's a very smart design to, if you look at the chemical structure here, is have 3 major is the 3 magic group is a mass reporter, mass normalized, and a mean reactive group. So the design just changes the composition of a heavy labeled nitrogen and carbon, so we can get a different version of the TMD tags. So the idea is labeling single cells in 384 wire plate, and each single cell we can label by unique TMT tags. So also Scuba MS use 200 cell as a carrier, so is to also minimize the sample loss and also increase the identification of the peptide, because 200 cells give very rich peptide fragment to help to identify this peptide. So currently that's the TMD 16 plex, so it means we can run 16 single cell in 1 hour LCMS. So, but there's still a significant challenge use this scope MS, because as you can see, all the single cells still prepare in individual wire plate. So there's still significant sample loss, and the digestion efficiency is also very low in the larger volume. That's why we try to combine nanopot with the scope MS. To design the research, we also realize the current nanopod design also have a limitation. So as you can see, we use 1 millimeter nanowires. It's still, compared with single cell, it's still too large. And so also we only can, because of not very large nanowires, we only can design 44 nanowires. It's a pretty low throughput for single cell analysis. And the very critical one is for TMT approach, we have to combine each set together. So maybe if you're working with a nanoliter dispenser, you may know it's pretty easy to put a nanoliter reagent to small vials, but it's really hard to withdraw them back. So that's a lot of technology challenging to do the TMT reaction combination for single cell proteomics if we use the nanopod. So to realize this challenge, we design new chip. We call it the nested nanopod chip. As you can see, the design is quite different from previous design. So we cluster 9 nanowires together and in one tight area, so that we can do one TMT set in one small cycle here. And as you can see here, we can get a very high density now. In this first design, we designed 243 chips. And a new design now, actually we are working now, is because there's a TMT16, we can design TMT 60 nanowires in one small cycles. So we can have we can run over 500 single cell in one chip now. So another challenge is because we are using much smaller nanowires, photophotometry cannot precisely sort the single cells into the small wires. So that's why we involve another cell sorting system. It's called Cellular1. It's use another robotic system to sort the single cell, not by the electrical field. As you can see, this is the first evaluation, we can reduce the cell size, the wire size to 0.3 millimeter. Actually, it's a similar scale with other microfluidic device. As you can see, even we use a much smaller 0.3 millimeter, we can still sort very precise sort single cell into the nanowires. Here is the SAM prep process. As you can see, we can sort the single cells, do all the SAM prep, and do the TMT dispensing, labeling. And the final step is to combine the TMT set. It's because of this unique design, so we can just simply add a 3 microliter droplet. So the droplet will just combine all the TMT labeled single cell sample together to the injection. So here is the evaluator with 3 cell type is epithelial cell, and raw cell is immune cell, and also endothelial cell SVEC. As you can see, we run 100 cells here. We totally identify over 2,400 proteins. And because there's a lot of missing values, so that's why we require 70% value values across all the cells. We still, even we require 70% value, we still can then quantify over 1,400 proteins from single cells. And as you can see here, the PCA, and it's pretty quite good separation of show the quantification quality of the platform. And we also try to, we always want to know what's the unique value of single cell proteomics compared with the single cell RNA sequencing. Is we are measuring proteins. So because we are measuring protein, we can actually then predict the cell memory marker. And we know it's for a lot of study, and we want to find out the surface marker for some unique rare populations. We can use for photophotometry to enrich them to follow up studies. So to do this, we match all the protein list to a surface protein database. So we get a list of this predict surface marker. As you can see, for each cell type, we have some predict marker. Actually, some are well known, like CD14 is the raw cell macrophage cell marker. So we also do immunohistochemistry to validate these markers, as you can see here. Very interesting for this surface marker, we can see actually they are enriched in the cell type. Actually, we can see they are enriched on the membrane. And also the rank of the infrared intensity is agree well with the rank of the single cell proteome measurement. Is again tell us the single proteome measurement is very precise, can measure the protein abundance in single cells. Also, we try to correlate the mRNA measurement, single cell sequencing measurement to single cell proteomics measurement. We use the 2 published data sets from the previous publication. We first did some similar studies, look at the periodic correlation for each modality. As you can see, for both cell types, the correlation of the RNA sequence usually at 0.6. Protein is pretty high, is almost 0.98. And so similarly, if we look at the coefficient of variations, it's also a way to look at the reproducibility of the measurement. For protein, we always get very low CVs. It means the model protein is give the very similar expression in the cultural cell line, but very different for the mRNA. They are quite different in different cells. is again tell us the very different measurement between the two approach. Here we also do some similar study to correlate RNA and the protein in each cell type. As you can see high correlation in the protein and low correlation in mRNA, and between them it's quite low, as you can see, usually below 0.3. Now, I'm moving to the topic of this the seminar today is spatial omics. So we know even we are very excited on the single cell proteomics, we do see that there's a lot of limitation of the single cell study. Is we have to rely on the tissue dissociation if we're working on the clinical sample. Also, we know is we lost all these very critical spatial information of the cell location or cell organization. And also very unique for protein is we know there are a lot of studies to show extracellular metric protein is very important for a lot of disease in the tissue. So if we do any single cell study for proteomics, we have to remove all these metric protein. That's why there's more interest to direct analyze the tissue to the spatial proteomics so we can look at the extracellular matrix proteins. So we did this by coupling a well-known laser capture microdissection or LCM or MMD, or use microscope and laser cutting to isolate the cells from the tissue sections. So to do this, actually, we use Zeiss. We use Zeiss because we have this one here. Also, it provides a adapter to allow us direct put our slide on the adapter, so we can direct collect the tissue from the LCM to our nanowire device. And to do this, we preload the chip with DMS droplet. The major reason use DMS droplet because actually it helps to capture the tissue direct and without loss them. Because if you know that is the up-down design. If there's no DMS droplet, they just fall down. So as you can see, we can add a low edge capture 200 micron size of the tissue and large at 200 into nanowires at here. So the first study, we always use the mouse brain because it have well-known substructures. And so we select 3 different area is the cortex, CC, and CP. We know that have different protein profiles. And so as you can see, we cut as first study, we cut 100 micron in diameter is a circle. So we can, usually we use a 10 micron thickness. So usually have a 10 to 20 cell in this each circle. As you can see here, we can get 1500 protein to 1800 protein from each samples. So we can quantify a half of them. The PC also tells us, you can see here, the protein profile across the 3 different locations quite different. Can tell us the high quantification performance of the platform. So we also try collaborate with Paul and Christine in our lab to do another, we call it the protein mapping. And it's just to collect all the tissue pixels in one region of interest. As you can see here, we just draw a grid here and dissect each square and collect them into the nanopod to the protein digestion. So after quantification, we can generate heat map, so we can overlay them with the tissue section. We can look at the distribution of the protein across the region of interest. So because for this tissue is the mouse uterine tissue, we know that measure 2 cell populations, 2 cell types. One is the luminal epithelial cells, as you can see here, and another one is stroma cells. So as you can see, this one actually we already did a statistical analysis to get this stroma enriched protein. As you can see, they are higher in stroma cells and low in the luminal epithelial cells. And typically, we can get a 2000 protein from these mouse tissues in each pixels. And if we can get here we can get several cell-specific protein profiles, and also that's also a value of spectral measurement is even in the same stroma cells we can see some kind of gradient from the top to down area. We can see a lot of protein actually have a similar trend. They're highly expressed on the bottom area of the tissue, have a low expression in the top areas. Even they're all stroma cells. So actually, I've talked with our collaborator. It's actually the oxygen gradient induced the microenvironment change. So it's the oxygen that enriched higher in the top area, the low enriched low oxygen level in the bottom area. That's why it induces the protein change. So this same previously we are using level 3, and so that's why we only look at the 24 pixels. Is because even the 24 pixels requires 3 days to run them. So that's why we coupling another, the single cell approach is the TMT labeling to multiplexing the analysis. So we call it the protein imaging with the nanopod, the TMT labeling. So we do the same things and do digestion and labeling and pull them for single MS analysis. You can see after the pulling, we can get pretty good protein. So we can get the 13 nanogram protein for each LC-MS runs. Even we use each pixel here is not 100 by 100, we use 50 by 50. So here, if we compare the performance of the TMT approach, and we reduce the size of pixel to 50 by 50 is 4 times smaller than level 3 approach. So we still get a similar protein coverage, close to 2,000 proteins. And if you look at how many time we require to run them, for LC, for level 3 we need almost 24 runs. Is almost 3 days. For the TMT approach, we need 7 hours. It's just more reasonable to analyze. So here also we look at the protein mapping result. For the typical protein, we know that enriched, here is smart emitter protein. I can see that, as you can see, that shows similar distribution between the 2 different measurements. And another protein is LOX15, is lipid metabolism proteins. We also can see they are enriched in luminal epithelial cells and in the 2 different measurements. And also very interesting, if we use a TMT, actually we can see they have a very low missing value. As you can see here, for TMT, we can see its protein in this pixel, in the pixel, but for the TMT approach, we can see all of them. So finally, I want to add several slides. Here is a new developing we are working on is a spatial result top-down proteomics. The reason we have interest because we know now we realize the protein in cell is not present in protein, actually present in protein forms. The reason because during the translation and also post-translation modifications, each protein have many different proteoforms. The predict of the protein form in each single cell type is 1 million of a protein form in one cell type. So it tells us if we want to actually understand the protein function, we have to directly measure the protein form, not the proteins. So if we look at the different protein form of the proteins, so the protein, so for typical for measure protein, we use the bottom approach. We have to digest the protein into small peptide. So we actually, because we digest small peptide, we don't know the combination of these PTMs. So if we protein have multiple modification, it's really hard to figure out where it comes from. That's why the very hot area in the proteomics, the top-down proteomics, to try to direct measure protein form use the mass spectrometer. So we did this last year to look at if we can measure small protein forms in low number of cells. As you can see, we get in our single cell is about 100 cells. We can measure 174 protein forms. We are very excited. It's actually demonstrate that we can use LC-MS coupled with NanoPOTS to direct measure protein form in small number of cells. To do this, actually, we realized the value of the nanopod top-down proteomics actually just to coupling with the mass spectrometry imaging, because we know mass spectrometry imaging actually can measure protein forms. But the challenge of the mass spectrometry imaging is that you don't know what the feature is. You only get the mass of the feature, but you don't know what the protein that is. So that's why we think maybe we can use a nanopod to give to measure the identify the protein forms, and then to help the interpret the multi-data. So here is a quick demonstration. Here is to we use a multiple red brain actually is we locate the two areas, the cortex and hypothalamus. So as you can see here, that we look at the two ketone protein. It's a modified ketone protein. As you can see, they have very interesting distribution in the mouse brain. And so we can use nanopod to identify these features to assign its ketone fold. And also not only the assignment, we also can do quantification to see actually the two measurements give the similar conclusion. There's very high in cortex for this protein and lower in hypothalamus area. And so, as we look at back how single cell sequence is developing in the 20 years, and it's pretty amazing how it's going fast. And I think single cell proteomics will follow similar trends. And I believe single cell proteomics may be still at the beginning, and you can see the coverage is still low, throughput still low, but we believe it will improve gradually in the next 10 years, 5 years. So for quick summary, we see we macrofluidic the nanopod can increase the overall sensitivity and to enable single cell and spatial proteomics. And we believe it can provide additional value for single cell biology, and it's still growing. We are very excited to see it, and we will believe we see progress every year. Finally, I want to thank, as you can see, it's not something done by one people in the lab. It's done by all the very tight collaboration between different groups in the lab, and it starts from collaboration. It starts from working with Rong Fan and Dick Smith to develop the platform. And finally, we're working with the cell isolation group, where Chrysler to integrate the facts and photocitometry, LCM together to do a spatial proteomics. And it's always collaborate with other group member to make the spatial proteomics and do all the cell isolation and chip fabrication. And finally, this is all the supporting from the NIH and DOE, and it's as you can see, the spatial proteomics major supported by HubbleMap. So I'm looking for questions. Thank you for your attention.
Thank you very much, Ying. It's really kind of nice tutorial as well for the audience to understand the history of the sort of single cell and the spatial proteomics. It's really discovery proteomics. I remember NIH was soliciting grant submission like a half a year ago. They were kind of sort of classifying the single cell proteomics into two different categories, and a very interesting number they put on the paper. So okay, if you're able to measure above 500 or below 500. So more than 500, that's defined as a discovery proteomics, and then below 500, that's targeted panel. Yeah, so I think the challenge in the field now is still sort of discovery scale proteomics, and the mass spec remains the main kind of powerhouse to drive in the field. So thanks. So I think already quite a few questions in the chat. I think Wenjun helped answer some questions, but if you want to look at the chat and further provide your own comments, that would be great. Also, I think people in the audience, if you want to ask questions directly, you can just go ahead and unmute yourself.
I think Ian has a question. Would you like to try? Otherwise, we can read those questions as well. Yeah.
So
okay.
Okay, yeah.
Oh, you've already read it, so you can, I guess, answer it yourself.
I'm looking for your questions. Yeah, just tell me your question. I don't know why
Just
here
wanted
I
to
only
know.
show several here.
Yeah, if you can hear me, I
Yeah.
was wondering if there's any way you can deplete abundant proteins, and if you think that that would help with the NanoPOTS throughput, you know.
For the for the LCM workflow, the deplete is always not always necessary because it's not a plasma. Plasma have very high dynamic range. That's why it's very challenging to measure plasma proton. For the typical single cells, actually the dynamic range is not a big challenge there. The major challenge actually is mass spectrometer sensitivity. If you look at a lot of bulk scale proteomics, actually they can measure 10,000 protein now. So it's a dynamic range of mass spectrometer is sufficient to measure all the proteins.
Mhm. And related to that also, when you show these clustered cells, the dense genes, will they be dominating your clustering algorithm? Or do you use like some normalization to, because some proteins, they could be 1,000 copies, some proteins would have millions of copies, right? So in the experiment, that it would kind of change the range.
Yeah, the way we measure the protein abundance is just look at the signal on mass spectrometer. So we get a signal of each peptide. So we roll up to protein level to get the protein abundance information. And for the normalization, actually, very good question. We do realize that we have to normalize the protein because as you may know, the protein mass spectrometer is a mass detector. The more mass you have, the more signal you have, right? So we see if we do not normalize the data, like we can you see big difference between larger cell and small cell. It's just simply not because the two cell is different, maybe just two cell size different, and just reduce the protein common number between large cell and small cell is totally different. That's why we usually use different kind of normalization, like media and also quanta normalization, make sure the two cell, assuming they have a similar distribution of the protein common number. Otherwise, it's very hard to compare the cell to cells.
Great, thank you.
Yeah, there's other questions. The protein group, yeah, this protein group actually is the way how we, I think we already answered this question. Is a lot of, we know protein have protein families, they have a share very similar sequencing, right? And sometimes mass spectrometer cannot, if we digest the peptide, digest the protein into small peptide, sometimes you don't know where this peptide belong to which protein. If we see such a problem, we just group all these proteins together as one protein group. So it's just a way to avoid artifact. We don't know which this peptide come from which protein. We just put them together as one protein group.
Thank you. That was my question.
Okay.
Can I ask you one more question?
Yeah, sure.
So it's amazing to detect 1,400 proteins from a single cell. So I don't want to focus too much on what you're missing because I know you're getting there. But I guess my question is, what kinds of protein, are there trends in the proteins that you're not catching? Like, are they membrane proteins or just hyper underexpressed or just hard to annotate?
So what we understand now is all based on the abundance. We only measure high abundant one. So our procedure now, we actually, we use a larger number of cells to evaluate if we have bias to only hydrophilic protein in some location of the cell. We know memory protein usually hard to detect, but we find out we do not have bias we don't even lose all this memory protein. If you look at the study, I look at some surface memory protein, right? In single cell, it's all there. So it's all matter of abundance. sensitivity limitation now. It's also actually we know sensitivity mass spectrometry is much higher. So that's why it's a complicated answer here. Is not only the abundance, also the sequence speed of mass spectrometer, because we have to measure one by one of the peptide. It's not like a single cell sequencing. We measure everything together, right? Use fluorescent imaging. For mass spectrometer, we measure one peptide by one peptide. So sometimes we have to compromise the throughput with the protein coverage, because we have to have enough time to measure each peptide. If we want to measure all the peptide, we need a longer time. That's why you compromise another throughput. So that's sometimes we can now we actually can measure 2,000 to 3,000 for some cells. We cannot do this if we want to measure all this, measure 100,000 cells. It's just the technology limitation now.
Thank you. Well, that's really awesome. Thanks for clarifying that.
Yeah. Do you have other question?
Yeah, I think you touch upon a little bit post-translational modification and I think one of those I think is significantly involved in the protein cell signaling network is the phosphoproteins, right? So are you able to kind of differentiate the phosphorylated versus total proteins, and even for a small subset of those proteins, because that can really teach you what the cells are doing, the protein signaling.
Yeah, actually, Wei Jun is an expert on phosphoproteomics, and actually he published a paper 3 years ago or 2 years. Wei Jun, maybe it's better for you to answer this question. I don't know.
So I think the phospho measurement is require even more sensitivity, right? So we sure right now, if we have a small cell population, say 1,000 cells, we can get pretty good data. But for the single cell phospho, it's still not there yet. So for example, in the Ying Zhu work, when he identified 1,400 protein, right? If we shoot phospho, maybe we can identify 100 phosphopeptide, but that will not be valuable in terms of quantification. So it's really a sensitivity limitation right now we are trying to overcome.
Yeah, for the analyzation, if we look at the phosphopeptide for the protein, they have phospho group, and that's why it's very hard to analyze it, because it's too hydrophilic. It's hard to get a charge, and so it's if you had 1,000 ions, maybe you only can collect 10 into the mass spectrometer. So that's a lot of challenge for phosphoproteomics at the single cell level.
So also related with the fragmentation of proteins, how is it an issue? I mean, is it really a problem, or you're identifying them like 100% accuracy?
So can you repeat your question?
The fragmentation of proteins, right? So when you read the mass spec, it fragments these chemical bonds and all that. So is that full protein, or is it like a subset of it?
For the fragment, we fragment the peptide. You mean the peptide, the sequence coverage, right? The question is sequence coverage.
Yeah,
it is not high. It's all depending on the abundance of the signal. Usually, I think it's 20% of the sequence coverage what we see now.
20%, you said?
Yeah, the 20%, the media, and majority of for single cell, actually majority of peptide, if you look at it, it's much lower. You only identify 2 or 3 peptides from each protein. And yeah, I see Mark have a very good question. It's on the question is TMT works well when all the sample in one set. If across multiple different set, it's have a challenge, because if we run 1,000 of single cells, we need at least 100 TMT set. And the challenge is each set actually is one batch. So here the batch is different from single cell RNA sequencing, because single cell RNA sequencing, one batch actually is 10,000 single cells. It's just one sequencing, right? one sequence runs in one chip. But for TMT-based single-cell proteomics, one batch is one
16.
LCMS runs. So that's actually the challenge in the field is realizing, but really where is the challenge we still not realized yet, because most of study now is working on the cultural cells, usually very low, only 2 or 3 populations. It's still working now, but really if we go to clinical sample, like if there's a 20-30 sample, 20-30 cell types, that's one challenge, because in each set, maybe the cell population is totally different. That's a very challenging to normalize them if we have this situation. So it's a really good question, and I don't think that the field have a very good answer to that. Yeah, maybe Weijun can provide more insight, because there's other large scale, not single cell, but also thousand samples have a similar challenge there.
Yeah, I think it's going to be a significant challenge for real application. Let's say we wanted to do 2,000 cell, and among the 2,000 cell, you have that 10 cell type or something. Right? But I think the technology has to be driven by many replicate of reference in order to help you to do the quality control and like the normalization, right? But I think people has to realize that single cell proteomics or proteomics in general is a combination of analytical measurement and computational power, right? And like the question a minute ago about the coverage, we never measure a protein. We only infer a protein exists or not based on computational algorithm. So basically our data never tell people this protein exists at 100% confidence, right? It's an influence. So like basically there's so many steps of computational. I think it's an interesting science area. I integrate many disciplines. Yeah.
Yes, the protein structure determinations, right? The Google project and all that. Actually, you predict the 3D shape and everything. Same challenge also occurs here, I think.
Yeah, that's bring up another, another actually good discussion. What we realize actually level 3 actually is better than TMT in terms of battery effect. Level 3 we run each cell in one LCMS runs, and what we see now is the level 3 is more precise because you don't have to worry about the battery effect. So also level 3 have a much better quantification, because the TMT, the major actually we all know is TMT is you isolate the peptide to the fragmentation to release the report ions. Actually, each isolation you isolate many peptides. That's why it's called co-isolation problem or retrocompression problem. So you see the signal is not from the same, not from one peptide actually. It's a signal from multiple peptides. So that's the second point of Mark is artificially inflate the sample to sample correlation. Yeah, Mark, are you here? I don't think I can. I know your question. What is exactly your question?
I appreciate it. You answered most of it, but if you're using TMT across a bunch of very different cell types, as you addressed, the only ones that would be, if you use like a control across all those samples, the only peptides you'd be able to see are the most abundant ones, which are probably ubiquitous, and you lose some of that cell specific detail. And that's going to make cells look more similar because they all have gap DH, but they may not all have the same
you only look at the
signature.
same population of protein.
Right.
So it's a really good question. And the field, actually, we know the challenge there, but we just do not have a good solution yet. But level 3 clear a good way to address this challenge. It's just level 3 is just very low throughput now.
Yeah.
Yeah, very good
agree.
question. Thank you.
I think label 3, it's a challenge with TMT across multiple cell types. I totally agree. Label 3 is probably better for that comparison at this point in time, but this will all change with the technology as it gets better.
Yeah, that's what we are doing here is, if we want to measure, less than 200 cell, we will avoid use level 3 here. And if we, over 500, there's no way to use the level 3 because it requires one mass spec for one project. Is no way to do this. So that's new question. It's from Eddie Wang. So I don't know. So my logic. I never heard this company. What's anyone knows?
I know this one. It's an antibody epitomer based like a profiling for plasma proteomics.
It's same as Olink.
It's a first generation company. Olink actually is outperform SomaLogic now.
Okay.
So Olink is like a antibody based. SomaLogic is epitomer based. Epitomer is a small
still like
sequence
a targeted panel. Just they have very large panel.
Yeah, so good thing is the panel is so large now. Right? They can profile nearly 4,000 to 6,000 protein in a tissue or maybe plasma.
Yeah, but I don't think they are getting to single cell now.
I know.
It's still way far from there.
Mainly limited to the biofluid, I think, for those technology. But if you want to do tissue or single cell, right, mass spec, you still have a significant advantage at this point. Yes.
And for spatial, the FFPE or fresh or different fixatives, do they make difference?
Huge difference. So yeah, we have a lot of trouble. We see protein from FFPE, and just the ID in the protein coverage much lower compared with fresh frozen. And so we still don't know why. It seems it's a mix of different problems. And after protein is cross-linked, it's some small molecule metabolite also cross-linked with the peptide. It just mess up everything. And I think the best result we get is 1,000 protein from 200 micron. The 200 by 200 micron. 1,000 protein is much lower than we can get from a fresh frozen tissue.
So it's basically like 100-fold less, almost.
It's like if you use fresh frozen, we can get 1,000 protein from 50 by 50, for example.
Oh, okay.
So to get the same protein coverage for FFP, we need 200 by 200. It's like almost 20 times larger sample.
One of the major problems of FFPE is proteomics measured protein sequence, peptide sequence, right? And FFPE as a fixation reagent really is a cross-linking reagent, right? Permanently alter the protein by itself. So even if we do get data, we still don't know what's going on with the FFPE reaction. So sometimes the data we got could be misleading. So so far we don't have enough data even to share it's confident in terms of quantification for FFPE.
Yeah, a lot of this actually we call is a chemical modification, right?
Yeah.
If a little bit of modification, you cannot identify it because you don't know what it is, right? Even you see all the peptide signal, yeah, you don't know what it is. So it's still challenging, but it's working somehow. It's just a very low performance for the spatial proteomics.
So I don't know if you did a D-cross-linking before you apply your kind of digestion in the nanopot.
We do. If we do not do D-cross-link, we see nothing. Okay. Like blank. Because first step we need to get the protein into the solution, right? So we do heating for very high temperature, use some kind of salt, right, buffer. So it helps, but still there's a lot of unknown modification after cross-link. That's actually very challenging for FFPE samples. So majority of your sequencing, in majority of the spectral, you don't know what they are. It's just Is maybe informatics can help some of them, like open search. You don't have to specify what's the modification. Maybe it help save some signal back. Yeah, it's challenging.
And would you happen to kind of maybe discover a new tag that can uniquely identify a barcode or something like that, right? Would that be possible in the future?
I think Thermo is working on this. Some officials have a huge interest in single cell proteomics, and every year they add 2 or 1. The 2 years they add from 11 to 16, and next year they add 16 to 18. Because the chemistry you never can get from 18 to 100, right? So we will see slowly every year. Maybe some will release maybe next year we'll see 20, 25 like that. I just don't know. That will have you have, unless there's a breakthrough in chemistry and synthesize all the isotope chemistry. I don't think it's very fast to get 100 barcode. Another challenge is actually it's an mass spectrometer, right? Because all the mass spectrometer have a limited charge capacity. If you push too many barcode inside, it cannot handle it. It have we call it special charging. So that's why there's a limitation on the barcode. So actually, I remember someone do some calculation. Seems it's fine to have 100 barcode. It's still doable to have 100 barcode. And 1000 actually is very challenging because the charge is not only, its charge is bring all this. If you put 1000 cells together, you have some limitation maybe on the special charge.
I see. Great. Any other questions? If not, I think let's thank to the Dr. Zhu and PNNL for this great presentation. I think it's clapping time. Let's use this reaction as well.
Yeah, that's a great discussion.
And before we wrap up, I'd like to also announce that next week we will host Dr. Michael Gerner from University of Washington, and he'll be talking about regulation of the immune response through microanatomy and cellular patterning. And with this, thank you guys, everyone, for participating today. And we'd like to hopefully see you guys next week. And please enjoy your weekend. Bye-bye.
Thank you. Bye.